Hi, in this guide, we are going to show how to do the clonality or diversity analysis in Platforma. So here I already have a project that we use for the VDJ guide. That's a project where we have bulk sequencing TCR and BCR data already pre-processed with the MyCR clonotype and log, and now we have our clonotypes available downstream to MyCR clonotyping. So to do the clonality or diversity analysis, we are going to use diversity analysis block. This is repertoire diversity. So let's add this block to the project. So once added, we have settings here. And in the settings, we have to select the abundance metric that we are going to use for the diversity estimation. And here we have a couple of options, and those options correspond to the immunological chains that are exported from the MyCR clonotyping block. Just to remind you, here in MyCR clonotyping, we have a receptors or immunological chains drop down where you can specify which immunological chains, in case of bulk sequencing data, or receptors were sequenced. And specifically for this type of chemistry, for this sequencing kit, all 7 chains were profiled simultaneously. So here I can select only one immunological chain, because for bulk data, different chains are amplified separately. Hence, you can do the diversity analysis, obviously for multiple chains simultaneously. So let's select, for example, immunoglobulin heavy as our abundance, and by the abundance, it means that the primary abundance metric for this clonotyping data will be used for the diversity analysis. And the primary abundance metric, which is used here, is actually defined by the preset. Well, specifically here, we use the selector driver map assay. And this assay, if we go to the MyCR documentation, so we go to the built-in presets, and here for the selector, for the selected chemistry, we know that this data, this version 1 of chemistry has UMIs. So the primary abundance metric in this case is a unique molecule count. So this unique molecule count for every clone type will be used as abundance metric. If our data does not have unique molecular identifiers, then the reads will be used here. So next you have here some defaults. Those are actually diversity or clonality metrics that will be calculated by default for this data. And let's go to the observed diversity, for example. So here we have a type, observed diversity. Observed diversity is just the number of clone types and sample. And one crucial parameter here is downsampling or normalization. So we have many samples in this project. Here we have many samples corresponding to different time points, and the goal of the analysis of the clonality analysis is to compare the clonality or diversity across different samples. And to do this comparison properly, we have to normalize our data. And this is pretty obvious because in different samples and different fibers, we took different amount of RNA, different number of cells, and then we amplify, etc., etc. So there is a bias, or we just have a different sequence and depths. Let's say for one sample, we took 1 million of reads in sequencing, and for another sample, which was sequenced like couple weeks or couple months after with different sequencing technology, etc., we took several millions of reads into the sequencing. So the proper downsampling or normalization is crucial for the statistical analysis of our data. And there are a couple of options here, depending on what type of analysis you are doing. Well, there is a none, meaning no downsampling at all. And for every sample, we will take all of the data, all of the clone types from that sample into the analysis with this non-parameter. Top N allows to pick just top N clonotypes from every sample. Cumulative top allows to set the percentage of the abundance that should be taken in the analysis. Let's say if we put number 80 here, it means that from every sample, the underlying tool will take that number of clonotypes that constitute 80% of top clonotypes, obviously, that constitute 80% of the total abundance of the sample. measured, as I mentioned, in this case, in terms of the UMIs. Or the option, which is the default, is random sampling. And this is a very powerful and the most, well, the standard way how you do this normalization. You just pick the randomly sampling that amount of sequencing data, in this case molecules, that will match the resulting abundance across all of the samples. So if you have one sample with 1 million of UMI, well, let's say, well, okay, 1 million, just for example, 1 million of UMIs, and another sample with, I don't know, 500,000 of UMIs across all of the clonotypes, then random sampling will apply a random pick, like randomly clonotypes and UMIs, not the clonotype, but UMIs, to have the same number of UMIs in all samples. So this is the most powerful thing. And with the auto setting, it will put the number of UMIs to which we downsample picked automatically from all of the samples. But we can use fixed, let's say, and select, let's say, I don't know, to downsample to the 100,000 of UMIs in each data set, or we can downsample to the minimal sample across all of the samples. Auto is our choice by default. Yeah. Now we have different metrics here. Shannon-Wiener diversity, Chow-1 estimate, Gini index, D50 diversity. Let's add, for example, normalized Shannon-Wiener index with random sampling as like additional option to the defaults. But as I mentioned, the defaults are already good. So let's click run, and it will actually trigger the analysis on the backend of Platforma. And now the only thing we have to do is just to wait when the analysis completes. It should be pretty fast for even for big data. Okay, very well. It was really fast. So here, once the analysis finishes, we have a table of all diversity metrics that were calculated. So for every sample that we have in the data, we have a columns with the diversity metrics that we have here in the settings. This is the table that we can export, but the real thing, the real interesting thing starts here in the diversity graph. And here we first time see the really powerful tool available in Platforma called Graph Maker. This tool, Graph Maker, allows to build a really comprehensive visualizations and bring many information on the plot and build like publication ready plots. So the first thing we see here is the data mapping. I click here on the data mapping, and we have, like, on the y-axis we have, let's say, the different things that we can visualize on, in this case, a bar plot. And let's, for example, visualize normalized Shannon-Wiener that we just calculated. Here we selected normalized Shannon-Wiener, we have it in the data, so we just select normalized Shannon-Wiener here. And now, as a primary grouping, by default, sample is used. But remember that in the very beginning of our project, we noted that bringing metadata associated with the samples is really crucial because it gives the context for the downstream analysis. And in this example, we have a time point as one of our metadata columns. And automatically, all of the data and metadata that exists in the upstream becomes available in the downstream analysis and visualizations. So here in the metadata in data mapping, I have time point, which I can now bring to the primary grouping. So now my samples are grouped by the time point, and we can play around with the type of visualization we use to explore this data. We can choose, for example, box plot or box plot plus jitter dots or violins, or the one that I like for the specific data is a line plus jitter dots. So now we'll really see some pattern, some nice thing that in the 8th day after the first immunization, this is specifics for this biological project, the diversity, the normalized Shannon-Wiener index, decreases significantly. Yeah, let's go further into this thing. So first thing we should note here that the actual time points are sorted on the x-axis alphabetically, and that's, well, we should change because the real time order is different compared to the alphabetical order or the words that we use for the metadata. And as you can note, I clicked on the axis settings, And in the access settings, we have primary groups order. Those are the primary groups. So let's click here. Well, the first thing, we can change the colors, which is already nice. And there are many options to change the colors. I like this one. And now you see the order of the points in the data. So I'm going to change this order to bring those primary groups in accordance with the real timeline. So that's what I'm doing right now. One, two, immunization one. So we have the control before the first immunization. Then we have the first immunization. We have multiple time points for the first immunization. Then we have actually something happened before the second immunization that's called Rx here. I'm not going deep into the biology context of the study. Yeah, so I brought this in the accordance with the time points, with the real timeline, and now the second immunization. Done. So now my groups here are in accordance with the real timeline. And yeah, we see the control before the first immunization, and then during the first immunization campaign, the diversity of our repertoire decreases, meaning that we have the clonal expansion and we have a clear signal of this clonal expansion. Let's make this plot bigger and nicer, and let's modify it further to bring more context here. And I'm going to use statistics tab. And for the statistics, well, this largely depends on the biological context of your study, but let's say we want to bring the overall p-value on the plot, Kruskal-Wallis, for example. Let's say we want to do a group comparison. So here at every time point, we have multiple samples, which is, well, this is a rich biological project. So let's say we use control before any immunization, control before the first immunization as our reference, and apply, well, again, let's say a Wilcoxon test or t-test, yeah, for all other groups to add the comparisons. And now you see those dots, you see the comparisons, and you see the significance. You can, well, play around here, and instead of significance, show the actual p-value. You can hide or show, make visible the non-significant comparisons here. You can also play with a pairwise comparison. That's largely depends again on your biological context, but let's say if you want to do a t-test across all of the groups, well, there are so many of them, but let's say if we do Wilcoxon, there are no any. Yeah, well, the statistics depends on the biology. Yeah, etc. So as you see, we can play around here to build and bring statistics on our plot. And this is very important that this statistics is really comprehensive. Let's say here we use a t-test, and we also apply automatically the correction method. We use a Bonferroni here, and this is, well, important to have the real statistics on the plot. So what else? Let's add the title to our plot. Let's call it normalized Shannon winner index. It's IG heavy. Yeah, I like this. IG heavy. Yeah, and yeah, that's it. There are other parameters how you can modify the shapes, the colors, and many different things. You can export this plot and just include it into your publication and do more things. The biology behind the clonality analysis is very important. So here I'm looking on the normalized Shannon winner index, but in the same way you can look at the observed diversity and other diversity indices that we have in the data calculated here, and this all depends on your biological context. In the documentation guide on our website, we elaborate in more details which diversity metrics, which clonality metrics are, well, should be used for different analysis cases. Thank you, and stay tuned for next chapters of our VDG analysis guides.